All right, let's dive into one of the biggest puzzles in all of modern biology. How do you actually build an organism's complete genetic blueprint right from scratch? This process is called de novo genome assembly, and trust me, it's like trying to solve a puzzle that has millions upon millions of tiny little pieces.
I want you to picture this. Imagine you have a massive encyclopedia, the whole set. Now imagine you run it through a shredder that creates millions of tiny overlapping confetti pieces. And then your job is to put it all back together perfectly. That is exactly what scientists are up against when they assemble a genome. See, our technology can't just read DNA from end to end, it can only read these tiny overlapping fragments. The real magic is figuring out how they all connect. And here's the kicker. It's not like we're just rereading a book we've seen before. We're putting it together for the very first time, with no idea what the original story even said. The whole goal is to take all those millions of short DNA sequences, we call them reads, and build them back into the complete continuous sequence of an organism's chromosome. But of course, it's never quite that simple, is it? The machines that read the DNA, well, they give us two very different kinds of clues, and each one has its own major pros and cons. This creates a classic scientific trade-off, and figuring out how to get around it has been a huge focus for years. So, on one side you have what are called short reads, and on the other you have long reads. You can think of them as two completely different types of clues for our shredded encyclopedia puzzle. Each one helps us in a totally unique way. So, your short reads are like finding these tiny, crystal clear snippets of text. I mean, they're incredibly accurate. Every single word is perfect. But the problem is, they're so small you have almost no idea where they fit in the grand scheme of things. Long reads, on the other hand, they're like finding a whole paragraph at once. They give you amazing context, amazing structure. The catch? The text is kind of blurry and full of typos. So you see the problem, right? The big question becomes, how can you get the best of both worlds? How do you combine that pinpoint accuracy of the short reads with the awesome structural information you get from the long reads? How do you make a genome that is both complete and accurate? And the solution? Well, it was a real aha moment for the field. It's called hybrid assembly. Instead of picking one method and sticking with it, scientists realized they could just combine them. You let the strengths of one completely make up for the weaknesses of the other. It's such an elegant solution, and honestly, it has revolutionized everything. The strategy itself is actually pretty simple, but so powerful. First, you use those long, blurry reads to build a basic framework of the genome, the scaffold. This gives you the big picture, showing you how all the major sections connect. Then, you bring in the millions of super accurate short reads to fill in all the fine details and correct all of those typos. So that's the big idea, but what does this actually look like in practice? Well, bioinformaticians follow a very clear blueprint, a kind of standard operating procedure, to turn all that raw data from the machines into a beautiful, complete genome. The whole process really breaks down into 4 core stages. First up is quality control. Then you have the assembly itself. After that, a really important step called polishing, which is all about fixing mistakes. And finally, assessment, where you see how well you actually did. Let's walk through each one. Okay, stage 1, quality control. You know that saying, garbage in, garbage out? Well, it is 100% true here. You just can't build a reliable genome with low-quality data. So the very first step is this rigorous quality check. Scientists use tools like FastQC and nanoplot to look at all those raw DNA reads and make sure they're good enough to use.
Now, before we get into the assembly part, we gotta know one key term, contig. A contig is just a continuous stretch of DNA that's been assembled. In our puzzle analogy, it's like a big chunk of the picture you've successfully glued together. The goal, of course, is to have as few and as large contigs as possible.
And now for the main event, assembly. This is where specialized software, like a program called Unicycler, takes on the massive task of fitting all the pieces together. It very cleverly uses those long reads to figure out the tricky, repetitive parts of a genome, which often totally confuse the short reads. It bridges the gaps and connects all the smaller contigs into much, much larger ones.
So, getting that first assembly is a huge step forward, but we are not done yet. Not even close. What we have is a fantastic first draft, but it's still full of those little errors inherited from the blurry long reads, and that's where polishing comes in.
You can think of polishing as digital proofreading, but on a massive scale. Software like Pylon will meticulously go through the entire draft genome, base by base, and use the overwhelming evidence from millions of those super-accurate short reads to find and fix any remaining errors. This gets the final sequence as close to perfect as we can possibly get it.
So, after all that work, we have our final, polished genome. It looks great, but how do we know if it's actually any good? The very last step in the whole process is a thorough assessment, where we formally grade our own work.
There are really three key signs of a high-quality assembly. First, you want a small number of contigs, ideally just one for each chromosome. Second, you want a high N50 value. That's a measure of how continuous the whole thing is. And finally, completeness. You have to make sure that it contains all the genes you'd expect to find.
Let's break down that N50 number, because it sounds really technical, but the idea is actually pretty simple. Imagine all your assembled contigs, all the big chunks of your puzzle. A high N50 value just means that at least half of your entire genome is contained within really, really large continuous pieces. It tells you your puzzle isn't just a bunch of tiny fragments, it's mostly big, beautiful sections.
But you know, numbers on their own don't tell the whole story. The ultimate test is a biological one. Does our final assembly actually contain the essential, non-negotiable genes that we expect to find in this type of organism? Because if key genes are missing, you know something went wrong along the way.
And there's a gold standard for this. A tool called BUSCO checks for a core set of genes that absolutely should be there. To be considered high quality, an assembly needs to contain more than 90% of them. This is the final thumbs up, confirming that our blueprint isn't just statistically sound, but also biologically functional.
So it really all comes back to this one core principle, which is just perfectly summed up in the standard procedure document. Hybrid assembly works so well because it leverages the high accuracy of short reads and the long-range continuity of long reads. It combines the best of both worlds.
And look, this is not just some theoretical exercise for fun. Assembling these high-quality genomes is absolutely revolutionizing our ability to understand genetic diseases, to develop crops that are more resilient, and to protect endangered species. It gives us the fundamental blueprint we need to start tackling some of the world's biggest challenges.
And that leaves us with a big final question. As this technology gets better and more accessible, we're going to have these incredibly detailed blueprints for all life on Earth. So with that kind of incredible power, what's the first problem we should tackle?